pleasure to welcome you to the Distinguished Faculty Lecture Series. Thanks for coming out this evening to celebrate the accomplishments of another one of our exceptional faculty members here at the University of Montana, among the many faculty members who are exceptional on this campus. My name is Royce Engstrom. I'm the Provost of the University of Montana. It's a pleasure to be here with you. This lecture series seeks to do really two things. One, to serve to recognize those members of the university's greatest asset, the faculty, and the individuals who make up that faculty and who have distinguished themselves in a variety of ways. Secondly, the purpose of this is to provide an exposure to a discipline that we may not have thought about before, and to provide an exposure to the excitement of that discipline and the significance of that discipline. And we hope you get that, we know you will get that in tonight's lecture. We have among our faculty here at the university people who are truly leaders on a worldwide basis in their fields. And at the same time, they are people who care deeply about their students. They're inspirational in their ability to tell their story and the story of their discipline and their research. For those of you who are students in the audience, and I'd like to just get a sense of that, would you raise your hand if you're a student of any level? Great. Thank you all for coming tonight. And I have one important piece of advice for you. If you can associate yourself with faculty members, the likes of the speaker tonight or any of the other speakers that we've had, and in fact any of our faculty members at the university, one of the best pieces of advice is to forge a good, close relationship with those individuals. They will be very influential throughout your lifetime. I still have very important connections to faculty members that I had in my first year as an undergraduate, and those people have been inspiring to me throughout my career. We began this lecture series this fall with Professor Jackie Moore, who is sitting over here. Jackie, raise your hand. Thank you for starting us off early in the fall. Jackie is our most recent Regents Professor, and she is from the School of Business. Second in the series was Dr. Garen Smith, Professor of Chemistry. I don't believe Garen is here tonight. And he was awarded the Case Montana Professor of the Year, and so he gave a good lecture a couple of weeks ago. Tonight is our final lecturer in this series for this year, and like the others, it will be immediately apparent to you why he was selected to present in this series. I'm exceptionally proud of these three faculty members, as I am the entire faculty at the university. Tonight's lecture is also part of the Climate Change lecture series. As you may know, we are among the first universities in the country to establish a formal academic program in the area of climate change studies. Just this last year, we received approval from the Board of Regents for a minor in climate change studies. And so we are delighted tonight that the Climate Change lecture series is a co-sponsor of this talk. We have several people in the room associated with that climate change minor, and so thank you for coming. So this will conclude the lecture series for this year. In the springtime, I hope that you attend the alumni lecture series, which will feature other of our faculty members here at the university. And of course, the presidential lecture series goes on all year, bringing distinguished guests from beyond the campus. Tonight, I'm pleased to call upon our Associate Provost for Graduate Education, Dr. Perry Brown, to introduce tonight's speaker. Perry is also the Dean of the College of Forestry and Conservation. He has been on loan really to us in the Provost's office this last year and the current year to strengthen our graduate offerings. So would you please welcome Perry Brown.
Well, good evening to all of you. This is a real pleasure to be here tonight to introduce our speaker. I had the fortune of being able to hire Scott Mills 15 years ago, and the decision to hire him has been reaffirmed nearly every day since then. He is an outstanding colleague and what I call a complete professor. He does it all, and he does it all with excellence. We honor him with this lecture this evening, partly because he was awarded a John Guggenheim Fellowship for 2009. But there is even more, as Scott is a professor who is being recognized and honored for the work he has done over many years. Early in his career, Scott was honored by the students of the University of Montana as the Silent Sentinel Professor of the Year for excellence in teaching. Also early in his tenure at UM, he was named an early faculty development awardee by the National Science Foundation for excellence in research and great promise for his future. He remains a top teacher and researcher and has added significant service accomplishments to his portfolio. Resulting from the outstanding work that he has done, Scott has testified in front of the U.S. Congress, served on committees for the National Science Foundation, the National Park Service, the U.S. Forest Service, the International Whaling Commission, and the National Marine Fisheries Service. He was on the Board of Governors for the Society of Conservation Biology, a member of the Western Governors Association Policy Working Group on Climate Change Effects and Wildlife, and was a contributor to the North American section of the reports of the International Panel on Climate Change. In addition, his work has been covered by many media outlets, including The Nature of Things with David Suzuki, Discovery Channel Canada, National Public Radio, Science News, Science, and many popular magazines and newspapers. Lastly, in introducing him, I want to make a few comments about his Guggenheim Award. Only a very small proportion of all people considered for Guggenheim Fellows are awarded one. And for 2009, Scott Mills was the only Montanan selected for a Guggenheim. Fellows are selected annually on the basis of impressive achievement in the past and exceptional promise for future accomplishment. Upon receiving the fellowship, Scott humbly indicated that it is an incredible honor that unexpectedly puts me in the rarefied air of the country's top scientists, scholars, and artists. It speaks highly of the world-class status we have at the University of Montana in the Wildlife Biology program and the College of Forestry and Conservation. A major focus of Scott's fellowship period will be helping to build capacity for ecological science and conservation planning in the Himalayan kingdom of Bhutan, where he will migrate with his family in January. To finish this introduction, I want to indicate that Scott has experience across the country and across the world. As one indication of this geographical breadth, he has degrees from North Carolina State University, Utah State University, and the University of California at Santa Cruz. Tonight, he will speak with us on the topic, Wildlife Biology in a Changing World. I give to you Scott Mills, an extraordinary professor.
Well, thank you, Dean Brown, for that kind introduction, and thank you, Provost Engstrom, for the kind introduction, and also for inviting me here to give this talk. I'm honored to be here tonight and appreciative for you all coming as well. 50 years ago, in 1948, a Swedish chemist received the Nobel Prize for figuring out that a particular synthesized chemical, an organochlorine, could kill mosquitoes by the millions. Also, he learned the same chemical was safe for humans and any other mammal. This was a wondrous discovery, promising to extend the lives of millions around the globe who face death from malaria and other mosquito-borne diseases. About 20 years later, as the production of this chemical was peaking at about 100 million kilograms being produced per year, two things happened. First, a young kid in North Carolina who was fascinated by nature and who loved being outdoors spent a lot of time on camping trips with his family. Sometimes they'd go to Park Service campgrounds on the coast. A typical highlight of these camping trips would be when a kid in the campground would yell, "The DDT truck is coming." And this boy and his brothers and every other kid in the campground would stop what they were doing and they would hop on their bikes or go running out. They'd all converge right behind the DDT truck as it went through the campground, spraying white fog. And they would just run behind the DDT truck, immediately behind it, in the fog, immersing themselves in that mysterious white fog, breathing deeply of the sweet, delightful, slightly pungent smell. This picture appeared 20 years before that in National Geographic, and it's a picture of kids on a beach in New York, also playing in the white cloud from behind the DDT truck. So it turns out that my brothers and I on the coast of North Carolina were not the first to enjoy this sport. Rather, it had been at least 2 decades that outdoor-loving kids in mosquito country had played behind the DDT truck, and society reveled in the contentment of mosquito reduction that came from this. Of course, I said there was 2 things that happened in the late 1960s. The second thing that happened was that biologists discovered that DDT was not soluble in water, but it was highly soluble in fats, so that it accumulated in the eggshells of predatory birds. At high concentrations, DDT weakened the shells of birds, so reproduction plummeted. Without reproduction, charismatic birds such as bald eagles, pelicans, and falcons screamed towards extinction. But in this case, society heeded the warnings of the biologists, and policymakers developed the Endangered Species Act. The Endangered Species Act actually was prompted by the endangered species that came from DDT. And at the same time, DDT was banned from production or banned from use in most of the world. After society made that commitment, biologists worked hard to learn about how to rear falcons and eagles and pelicans and other species in captivity, how to reintroduce them and how to recover them. And so the really great news is that every species that was listed by the Endangered Species Act due to DDT is now either fully recovered and delisted or very close to being so. So that experience where biological science was used to both raise awareness of change, of a problem, and also to solve it was very influential for me as a kid. And so now I have the greatest job in the world because I get to learn, I get to get paid to figure out how nature works, and then think about how to convey that knowledge not only to students, but also to apply it in the real world to land managers, politicians, and the public. So what I'd like to talk about today are some of the changes that I've studied as a wildlife biologist and some of the zigzags that brought me here. And I guess actually for truth in advertising, in case people do need to bail, if people came here for a talk purely on climate change, it's not going to happen. And that's kind of intentional. It's partly because, as I'll talk about, I will talk a bit about climate change, and I think that climate change effects on wildlife is one important stressor or perturbation that we should be paying attention to. But I also feel strongly that there's lots of other things that we should be thinking about. And so for me, thinking about effects of change on wildlife is not just one of climate change, it's lots of other things. So I'll talk a little bit about climate change, but lots about other kinds of change. And I guess there's a point for that, the point for my doing that. This is where it started for me. My first experience in wildlife science was as a volunteer in Olympic National Park. I got to work in the backcountry all summer long, and as I did that, I got to learn the challenges of doing science in remote areas, and I also got to experience the joys of the sorts of things that scientists did. But more importantly, I was struck by the fact, I was struck by the role that science had to play in the societal decision of what to do with this invasive species. It's hard to believe because mountain goats are so remarkable and they seem so at home in the mountains of Olympic National Park, but actually they're not native there. And the plants in the high country of Olympic National Park are not evolved to withstand mountain goat grazing and wallowing. Mountain goats were introduced in the late 1920s before it was a national park, and then by the time it became a national park, mountain goats were increasing at 10% per year, and so that by 1980, there were more than 1,000 mountain goats. There'd been biological research going on for 2 decades. It was still going on when I was there. And the research had shown unequivocally that while goats had prospered, at least 33 known plant species, many found nowhere else on the planet, were being hammered by the goats grazing and wallowing. So what to do? Allow the non-native but very cute mountain goat to drive to extinction plants that are found nowhere else in the world? Come in and live remove these mountain goats and try to ship them off to somewhere else, maybe where they're not native somewhere else? Come in with helicopter gunships and kill the mountain goats in a national park? Well, it was clear to me and very moving to me that the decision that would be made on this would obviously be made based on society value judgments. But what really excited me was the role that biological science would have to play in helping to make that decision. The decision wouldn't be made by biologists, but biologists would be asked, how many goats are there? What's their trend? Are they increasing or decreasing? What are their effects? What's the best way to reduce their numbers? What happens when we start to reduce their numbers? So that experience was enough to cause me to switch from being a pre-vet major to being a zoology major interested in wildlife biology. And after playing around for a year or so, I then went to graduate school, and my research project in graduate school was a fairly brutal field project, sort of a 15-hour-a-day, 7-day-a-week sort of field work, which is a lot of what our business is about, so it's good to learn that early. But my project used radio telemetry to learn how the home range size and movement patterns of coyotes were affected by the numbers of their primary prey, black-tailed jackrabbits. So in the end, we learned how coyotes respond to drastic changes in their native prey, and that got fed into discussions of how to minimize livestock depredation in years where coyotes were predominantly territorial versus years where they were non-territorial and much more transient. Well, after this master's project on coyotes, I spent a 6-month stint in Washington, D.C., working with the National Wildlife Federation to learn how science interfaced with politics and policy. And then I bailed out of that and took off the tie and the suit and went back to Pacific National Park to work as a biologist in charge of spotted owl research immediately after they'd been listed as a threatened species. Soon after that, I headed back to graduate school, and the project that I came up with for my dissertation addressed the effects of forest fragmentation. This seems like a really well-known change or perturbation now, but at the time, very little work had been done, very little study had been done to understand the effects of this particular change on wildlife species. So my research project was designed to ask how forest fragmentation would alter both numbers and interactions among species. For this project, I decided to go small, and I chose to focus on small mammals that had strong interactions with their community and that potentially would respond to this kind of change or perturbation in different ways. The first species that I focused on is the California redback vole. This is in the genus Clethrionomus. This is not your garden variety vole. This is not the vole that eats the roots of your apple trees or that gets into your garden. Those are nasty microtus voles. I'm just kidding. Those are microtus voles. These are a different genus of vole, and they're mycophagous. They eat fungi. About 90% of their diet is the fruiting bodies of below-ground fruiting mycorrhizal fungi. So as you know, when mycorrhizal fungi fruit above ground, they are called mushrooms. When mycorrhizal fungi fruit below ground, they're called truffles. And almost the entire diet of this species is truffles. By the way, there's a little radio collar that I figured out how to put on these guys. We're also studying how they use coarse woody debris or logs on the ground. And so this was automatically an interesting species for me because without mycorrhizal fungi, there are no trees. So trees are inefficient at obtaining water and nutrients, sorry for people over here, I only have one pointer. Maybe I'll switch. The trees require mycorrhizal fungi because what mycorrhizal fungi hook into the roots of trees, and then trees are able to obtain water and nutrients through the network of hyphae that mycorrhizal fungi send throughout the forest. In exchange, the mycorrhizal fungi get their groceries. They get photosynthate from the trees. So this is a very tight symbiotic relationship between the trees and the mycorrhizal fungi. Well, how do the mycorrhizal fungi that fruit below ground disperse their spores? They can't do it through the wind and the rain like mushrooms do. So they rely on being eaten by animals and then having the spores defecated out in different areas. And red-back voles is one of the primary dispersers of these spores. And so clearly the red-back vole had a big effect on the trees, a big positive effect on trees regenerating. But the question is, what was the effect of removing trees on the red-back vole? And so that was one of the things we looked at. And what I'm showing here is the density of voles. And the first bar is the density of voles on clear cuts. And you see that voles are very rare on clear cuts. The last bar is the density of voles on control areas, unfragmented areas. And you can see they have high densities there. And then on the fragments, the forest fragments surrounded by clear cuts, voles are also present, with more voles being found in the interior of the fragments and fewer being found near the edge. So this was a bit of a surprising, non-intuitive discovery of this particular form of change, that logging could actually end up impeding the regeneration of forests on the logged area by inhibiting the mycorrhizal fungi, which then inhibited the vole coming in to disperse the mycorrhizal fungi spores. Now, one of the points I want to make about change, and again, I could do it with climate change, and actually I will do it with climate change later in the talk, but more broadly, anytime we talk about effects of any change on wildlife, it's really important to not think about change as being some monolithic force that's going to affect all wildlife species in the same way. I always bristle when people say, what's the effect of something on wildlife? Because I say, well, what do you mean? Do you mean a newt? Do you mean a whale? Do you mean a falcon? What do you mean? And so in this case, a species that was very positively affected is the deer mouse. Deer mouse reached enormous numbers on the clear cuts and on the forest fragments. And deer mice also are voracious seed predators. And the interesting story that emerged from this was that trillium, which you see down here in the bottom left, and which people here probably know about because we have quite a bit of trillium here in western Montana. Trillium, it turns out, live to be as old as we do. They live to be about 70 plus years old. And on these forest fragments, it turns out that the trillium that are on the forest fragments are all older than the age since the clear cut surrounded the fragments. So what's happening is that these these deer mice increase in numbers. They eat all kinds of seeds, annual and perennial plants, including trillium seeds. And basically, no trillium seeds make it past the deer mice in order to recruit as young trillium. So there's still trillium on the forest fragments. They're just getting older and older and older until the deer mouse numbers end up going back down and the seeds are released in order to germinate. So a different response by different wildlife species to the change or to this perturbation. And again, a bit of an unexpected cascading effect of a change in a wildlife species on other members of the community. So the trillium are getting very old. All right, well, it turns out that my PhD dissertation was at the right place at the right time. Two amazing things were happening in biological sciences. One was that the availability of computing power, of desktop computing power, had been fully embraced into sciences, really all the sciences. But what that meant in biological sciences is that there was now incredible developments in computational, statistical, mathematical approaches to actually be able to understand complex population dynamics and complex community dynamics. In short, we had much better quantitative tools to understand the complex things that happened in wildlife biology. The other big development that was happening exactly at the same time were big advances in genetic analysis. And with your permission, I will not talk about any of the quantitative stuff, but I will talk a little bit about the genetic analysis. Although both of them I've been very excited to continue using throughout my career. The thing you need to know about genetic analysis is before this time, if you wanted to know anything about genetic structure of a wild animal, you had to do two things. First of all, well, maybe three things. First of all, you had to go out and kill it. Second of all, you had to pull out its brain, heart, liver, or other protein-rich organ, because those were the only genetic techniques that were available to use. And third, you had to end up using genetic techniques that were pretty coarse. They just looked at the protein products, and they didn't really have high resolution to be able to understand subtle changes in genetic variation that came from recent changes due to humans. But that all changed with the invention and the widespread availability of the polymerase chain reaction. So the polymerase chain reaction allowed scientists to actually look at the DNA. Amazing, to look at the DNA, and furthermore to look at DNA from very small, very old, very degraded samples. And I just want to really briefly sort of say, and for those that don't know what polymerase chain reaction is, because it is now so fundamental in so many fields, I mean, in a nutshell, you isolate DNA, and it's okay if you only can get a small amount of DNA, and then you basically photocopy it. You break apart the double strands of DNA, and then, this part blows me away, you add into the tube the building blocks of life. You add in nucleotides, adenine, thymine, cytosine, guanine. You just buy them from Fisher, and you dump them in the tube. It just blows me away. I mean, all life on planet Earth, that's all they have, are those nucleotides, and there you are, dumping them in the tube. And then these primers attach, and basically what happens is the building blocks of life attach to the broken strands, and you've just doubled your number of DNA. You keep doing that, splitting the strands and photocopying them, and you can get a billion or more fold increase in your amount of template DNA. So this blew apart our world. It opened up the world of historic and non-invasive sampling. Now we could ask, what was genetic variation like way back when? What was it like 10 years ago? What was it like 1,000, 10,000 years ago? And how does it compare to now? And then it also let us obtain DNA, genetic variation, assay genetic variation in samples of very low quality and low quantity. It let us sample animals that didn't like to be sampled. We could find anything they might leave behind. Any little scrap that they might leave behind, we could find it, and we could learn things about the animal. All we need is a bit of blood or hairs or urine. I have to always laugh. I have to tell you this side story. Sloughed skin, like dandruff works. And I was at a whale meeting one time, and I asked this guy, the whale people were the first ones to use non-invasive DNA sampling to estimate population size. And in their scientific paper, they say they use sloughed skin to get a DNA sample from it. And I was talking to the guy that published that paper one time, and I said, so Scott, I said, that sloughed skin, that's basically dandruff, right? And he said, yeah. I said, what's that like for a humpback whale? And he said, well, they come up and they splash back down and they leave their dandruff behind. And I said, what's dandruff look like? He's like a piece of sheetrock. So that's whale dandruff. And so since 1990, I've embraced these genetic methods and the quantitative methods, but we're staying away from those, combining those with field studies to understand wildlife populations. And what I'd like to do is briefly talk about 5 examples of doing this. First, go back to our old friends, the redback voles, and remember they had high numbers on fragments, but remember they had very low numbers on the clear cuts surrounding the fragments. So the question is are they isolated? Are the fragments like an island and the clear cuts like an ocean, and are these animals losing genetic variation by being on these fragments, by being isolated? And if we measured heterozygosity, which is on the y-axis here, which is a measure of genetic variation, and we did it for 2 fragments and 2 controls, and you can see that there's just no difference. And so the conclusion here is that it's good news that the voles are maintaining their genetic integrity despite population fragmentation. Actually, I guess while I'm wired now, I can move around a little bit. The next question that I wanted to look at is actually one that we continue to look at now with lots of species, and it's a big issue in our field, and that is, to what extent can we estimate abundance using genetic sampling? The problem is when we want to know abundance of an animal population would seem to be a really easy thing. It would seem like you could just go out and count them. But the problem is that you don't see them all. And so what you've got to do in order to estimate the numbers of animals in a population is you've got to statistically come up with an estimate that involves having individually marked animals and then releasing them and then recapturing them and using the patterns of marked and unmarked animals whose identities you know to come up with a statistically rigorous estimate of abundance. And so this requires a lot of work. You've got to use traps. In this example, we've got a snowshoe hare and we've got a trap. We need about 80 of these traps to get a good statistically rigorous estimate of population size. And so if we're working in Glacier National Park, which we were on this project, it's a lot of effort to haul 80 of these things into the back country in order to estimate abundance. And so the question is, are you better off taking a non-invasive genetic sampling approach, where you just take some pieces of landscape cloth, you take a little bit of bait, you put the bait in the middle of the landscape cloth, you leave it out for 4 days, some hares hop along, they eat the alfalfa, they poop on the landscape cloth, and you can then obtain genotypes out of the feces, and you can use the individual genotypes to set up that individual mark recapture stream that you could use to estimate abundance. And so it turns out that if you do the genetic sampling at low hare densities and in remote backcountry areas, non-invasive genetic sampling is actually a more efficient and equally accurate method for estimating abundance. So here we'll switch gears, but still continue with this theme of applying genetic methods along with non-genetic methods. My student, Tammy Mildenstein, has been interested in 2 species of fruit bats in the Philippines. These are amazing creatures. They have wingspans of about 6 feet. They're often called flying foxes. They roost together, these 2 species do. It's hard to tell them apart unless you really are very careful. But one species is endemic and declining, heading precipitously downwards. The other species is prospering. Hard to tell them apart, and the questions are, why is it that one species is doing so well and the other is not? And so Tammy's already used radiotelemetry to understand something about where these animals go. Now she's collecting feces from underneath these roosts where both species roost, and she's using that to try to understand how many of them are there now, how many of them have there been historically, how much are they moving between the different islands in the Philippines. The last 2 stories of this about genetic methods that I'd like to talk about both have to do with the Canada lynx, a species which we know well here in western Montana. And the first story has to do with the simple question of where are they distributed across the United States. Of course, lynx were listed in the year 2000, and even when they were listed as a threatened species, nobody really knew where they lived, which is a bit discomforting to not even know where they are, much less how many of them there are. And the reason for that, of course, is that lynx, like many large carnivores, are just darn hard to sample. They're elusive, they don't want to be caught, and they're at relatively low densities. And so on this project, I worked with Kevin McKelvey and his group at the Rocky Mountain Research Station with the Forest Service here in town. And these guys developed a method of obtaining hair from lynx. So this is the hair pad. We had 22,000 of these across the northern United States. It's a 4x4-inch piece of carpet with nails through it, and it's sprinkled with bait, and it's nailed knee-high. You can see it there on the tree. That's the pad. And the idea is that lynx either see this visual attractant, the pie pan, an old trapper trick, or they smell it, and then they come up and they just do what cats do. They just rub up against it, and when they do, they leave hairs behind. And when they leave their hairs behind, we then developed a diagnostic DNA test to be able to tell what species left the hair. is it a lynx, bobcat, cougar, grizzly bear, or what? And this was successful. We put out more than 20,000 of these rub pads. A lot of people were involved in this, people all over the northern United States, putting these things out, 13 states over 3 years, and we learned a lot about where lynx are distributed. This also is, though, there's more to this story, which I suppose reinforces the importance of high-quality science and the interest of society in wildlife science. And this is the one that sent me to Congress. Perry mentioned that. What happened was some guys in Washington and Oregon took it upon themselves to mislabel some samples that they pulled off of a wall mount and off of a petting zoo, and they labeled them as if they were collected out in the field. And so a huge political brouhaha blew up. And I won't go into it, except for I'll say that it sort of culminated in this testimony to Congress, and I think it ended in a really positive way. It ended with Congress, I think, being convinced that indeed what these guys did was wrong by any standard. What these guys did violated any ethical standard that scientists should have in terms of handling the integrity of the data. On the other hand, it also the integrity of the study overall, and particularly the use of non-invasive sampling to understand the distribution of wide-ranging species, was upheld. So neither side of the political spectrum was really happy with that result, but I think it was the right one to say that what these folks did was wrong, to mislabel samples, but also the integrity of the study was upheld. Okay, the last one I want to just mention briefly, Mike Schwartz was working on a very different Canada lynx project with very different sorts of sampling, but the question here was, actually I'll go back, the question here was how much are lynx moving across western North America? Nobody knew. Why? Because lynx are secretive and elusive and don't like to get caught, and so we didn't know how much they moved. So we used genetic methods, and what this graph is basically the y-axis is just a measure of how genetically different are two populations. And we're looking across the x-axis of kilometers. And so what this says, it's just a cloud, which just says that whether populations are close together or far apart, they have sort of the same amount of genetic differentiation, which tells us that lynx are moving a lot. Lynx are moving a lot across big distances in order to prevent that genetic differentiation from happening. And so this was very important had very important sort of conservation and recovery implications, because it said that we shouldn't be focusing all of our efforts on just the places where lynx are right now, because lynx population dynamics may well be determined in part by the movement of lynx coming down from Canada. So it really established this importance of connectivity for lynx persistence and recovery. Okay, so some of you are like, when is the climate change starting? I'm going to get a D. Well, I do want to talk a little bit about adaptation to climate change. And again, I wanted to set this up as, sort of pushing the idea that there's lots of kinds of change in the world, and climate change is definitely a big league one, but it's certainly not the only one. And then I want to very briefly also talk about the work that I will be doing coming up soon in Bhutan. So we actually know a lot about climate change. I always tell people, I get them nervous by saying, actually, I can tell you exactly what will happen to every wildlife species, 1.5 million eukaryotic species on Earth. I know what will happen to them under climate change. I do. They're going to do one of these three things. They're either going to move to somewhere else, and there's actually been lots of cases of animals moving northward, or moving upward in elevation, for example. Either species will move, or they'll deal with it in place. They'll either, they'll withstand the changes and stay in place and still be there, or they won't do either of the first two, in which case they'll go away. They'll decline towards extinction. And that's pretty much the options for what will happen. So we know quite a bit about the move part, but the interesting thing is that we know very little about the capacity to adapt to climate change. I mean, we know that adaptation could occur through a couple different ways. We know that species could just deal with it within the envelope of the morphology and the physiology and the biochemical pathways they have. They might just make little changes within their processing, like gardeners talk about acclimation, where a plant becomes maybe cold tolerant during the year. So that could happen. No change in genotype, no change in gene frequencies, just dealing with it. But also we know that any kind of change, including climate change, exerts a selective gradient to which animals may respond by changing gene frequencies and evolving. So evolution by natural selection is certainly an option as well. Thing is, we've never really been able to crack this nut because it's really tough. You've got to come up with a climate driver. You've got to connect that climate driver to a specific phenotype, a specific form of an organism, and then you've got to establish what are the linkages between those two. What are the genetic, physiological linkages that cause the climate driver to change the phenotype? You've really got to do all those things to understand the extent to which species may be able to adapt to climate change. But I think we've got a good system to do it in. I've been studying snowshoe hares for about 10 years. Most of my work has been on population dynamics and response to different kinds of logging regimes. But one thing you see when you study snowshoe hares, you see a lot of this, and I'm sure a lot of you have seen this too, living here in western Montana. You see that hares in the summertime are brown, and they're pretty well matched to their background, and hares in the middle of winter are white, and they're pretty well matched to the background. The interesting thing is that the driver of this color change from brown to white has nothing to do with whether or not there's snow on the ground. It's driven entirely by photoperiod, by day length, independent of the presence of snow, which is very interesting given that the number one strongest, most predictable, most pronounced physical signal of climate change throughout the world in temperate regions is a decrease in the number of days with snow on the ground. This is out of Steve Running's shop and for our part of the world, and the main thing to notice here is that everywhere that it's blue, there's no red on this map. And so that says that everywhere that there's been a change in the snow melt, the timing of the snow melt in the last 50 years, it's always earlier. So snow melt's coming earlier and earlier. In the fall, it's coming, the snow is arriving later and later. This is happening in Europe, where the number of days with snow on the ground is a month shorter. It's happening in the Himalayas. It's happening in the Pacific Northwest. It's a very unequivocal signal. And so we're going to have a lot more of this. These neon hares, I like to call them, these white hares hopping around on a dark background. And you've got to realize that a snowshoe hare is lunch for every single creature that eats meat out in the forest. Raptors eat them. Any kind of meat eater, pretty much bigger than a shrew, eats snowshoe hares. And so, it's kind of almost embarrassing, when we're doing field work and you walk along with telemetry and you see a hare like this sitting real still, acting like it's hidden. You kind of just feel like you've walked in on somebody in the shower. You kind of want to look away, and you're kind of embarrassed for them. But the question is what's going to happen? What is the cost of that mismatch? How much will survival be affected? How much will population dynamics be affected? And then equally important, possibly more important, is this question of adaptation. Will snowshoe hares be able to adjust the timing of their color molt through phenotypic plasticity and or evolutionary change to adapt to climate change? And I want to really emphasize that that could be the case. This is not at all, in my opinion, a gloom sort of doomsday scenario. There's lots of reasons to think that hares actually may be able to adapt and that other animals may be able to adapt, in this case, the timing of color molt. But to do it, we're going to need to line up all those pieces. So I've brought in lots of help from the UM campus here. Steve Running's going to help with the snow downscaling models. Doug Emlin's going to help measuring selection. Kriya Bruner with determining the physiological mechanism of the coat color change. And Jeff Good, who's not even on campus yet, is going to help. He's a new hire, is going to help figure out the genes that are causing these changes. And I just wanted to show you this. This is just fun. I had a few radio collars left over from a previous project. So this fall, we've been going out and every week taking pictures of hares to look at their coat color and also the background to see if they're mismatched. And so this is hare 1333, middle of September, completely brown. A couple weeks later, you can see a little bit of white here, lightening up, and white in the ears. We were setting traps to catch other animals to put radio collars, so sometimes we caught animals that were already radioed. That's why these are in traps. But you can see a little bit more white and lightening up here. By October 24th, you can see it's substantially white. October 30th, there's just a little bit of brown left on top of the head. Otherwise, it's light gray. And this past weekend, this is indeed a white hare on what was this weekend. It was just sprinkling snow, but not snowing constantly on a dark background. So what will happen? Well, we've got to figure out the physiological mechanism. I'm not going to go through this. I'm going to just say this was developed by Kriya Brunner to sort of think about how the mechanism might work. I wanted to say that it's amazing. Nobody has studied this in any color-changing animal. Ptarmigan, you may know, change. Arctic foxes change colors. Weasels change from brown to white. Some species of lemmings change from brown to white. Lots of lepus, hare species, change from brown to white. And nobody knows how it works or what are the consequences of mismatch. So we don't know what the mechanism is, but our best guess is this, and I won't go through it except for to just say that hormones like melatonin, which many of you know is released more as the day gets longer. Melatonin affects melanocytes, pigment directly. It also initiates cascades of different hormones that turn on and turn off and end up producing brown or white hairs. So this is the world we're going to be drilling into in the next couple of years, we hope. It's very interesting, as I was writing this grant, to find Newsweek magazine cover it. This is our project they're talking about here. And they do talk about our project. But the thing is the article itself was pretty good. The article conveyed this point that hares may be able to adapt and they may not. The title, though, was a little, I thought, well, I guess they're, it's a title, right? So they make it seem pretty extreme, that depend on the fate of the vanishing snowshoe hare. Well, we don't know that they're vanishing, and in fact, we're just starting this research project. And of course, hares are not rabbits. There are no seasonally coat color changing rabbits. Hares are not rabbits. So my answer to that is maybe, maybe not. I mean, for sure they're not rabbits, but maybe they're disappearing, and maybe they're not, and maybe they'll adapt, and maybe they won't. All right, one other little bit of change on this topic of change, though. Remember I said how change will favor some species and not favor others, and this is a study we did back to the Olympics for me. I figure I have the ideal job. I get to live in Missoula and go do research sometimes in Olympic National Park. These are amazing creatures, these marmots in Olympic National Park. They have a different number of chromosomes than any other marmot species. So they're truly different species. And you would think these would be prime candidates to be affected by climate change. After all, they're on a peninsula. They can't move north because they'd move into the Puget Sound. And they rely on snow covering their burrows when they hibernate for 7 months out of the year. But it turns out that we threw lots and lots of stuff at them. I mean, we did genetic studies, we did marker capture studies, we implanted with radiotelemetry. We learned a lot about marmots. And what we learned was that they are declining precipitously, and it has nothing to do with climate change. Do you see what it has to do with? Coyotes. Coyotes have just invaded the high country of Olympic National Park in the last 20 years, and they are clobbering these marmots. Why? It goes back, this was very wild for me, because it goes back to that time as a sophomore when I was watching those mountain goats in Olympic National Park and thinking, wow, these plants don't have a prayer making it against this mountain goat to which they've never seen in their evolutionary history. Well, it's exactly the same thing here. Marmots have never seen coyotes. They don't really have any terrestrial predators that have eaten them over their history. And so they just are clueless, kind of like this guy. They just get really hit hard by coyote predation. So once again, here comes the societal value judgment. What do we do about this? Do we let an invasive species drive an endemic species to extinction? Do we kill something in a national park? It's a value judgment of society, but it's very exciting to see that wildlife biology will help inform this. We actually know now from our last few years a lot about the marmot, and also we've done a lot of work with the coyotes. Okay, the last thing, in just the last couple of minutes, I want to just briefly mention is exciting opportunity that's coming up in the next few months, which is basically for me an opportunity to think, can I take everything that we've learned in sort of in our field in wildlife and conservation biology? Can we take all of these techniques and apply them in a meaningful way in Bhutan? So I want to especially thank Sewang Wangchuk, who's doing his PhD with me using non-invasive sampling of snow leopards, and Timpa, who's now finished his master's and is now the first faculty member in wildlife and conservation biology in Bhutan. They are responsible for drawing me into this and also for providing most of these beautiful pictures I'm going to show coming up. Bhutan is a tiny little country sandwiched between China and India. It's 1/8 the size of Montana, but it goes from the lowland plains that are adjacent to India all the way up to the high Himalayan peaks, all in this one tiny country. The country is predisposed towards conservation. It's a Buddhist is the predominant religion, and a reverence for all life is very strong. And as a result, and as sort of an indicator of that, the country is 3/4 forested, with 1/3 of it in either protected areas, formal protected areas, or biological corridors. And the biodiversity is just phenomenal. Elephants, langurs, tigers, snow leopards, this one in a tree, takin, the national animal of Bhutan. Remember I said it was 1/8 the size of Montana, and yet there's more mammal species than Montana. There's 3 times as many bird species, and they're finding new species all the time there. So truly a remarkable biodiverse place with a commitment to keep the biodiversity in place, but also a desire to modernize. The country has recently gone from a monarchy, very peacefully gone from a monarchy to a parliamentary democracy, and they're very interested in modernizing. That includes urbanization, it includes hydropower, it includes mining, potentially it includes logging. It includes doing these things, but they want to do it with an eye towards keeping the biodiversity that they have, the remarkable biodiversity they have. And so, of course, there's lots of ways of helping to achieve that. And one way is to help build local capacity, the ability for local folks to be able to conduct wildlife science so that they know how many animals there are, how well they're doing, how they're responding to changes, and what risks they might have. Okay, so through this talk, I've only barely touched on the kinds of issues that we work on here in the wildlife biology program. In this slide, I have just 1 week's worth of clippings from the Missoulian that have to do with issues that we work on in the wildlife biology program, the students and the faculty. I also snuck in some from that obscure East Coast paper that are directly talking about the wildlife biology program here at University of Montana. And the reason I wanted to put these in here is just to say that obviously these kinds of issues are of interest to society, and there's lots of questions. But the point that I want to make is that the answers to these questions do not usually emerge from intuition or common sense or emotion, no matter how fervently some people wish that they would emerge from those things. They come from science that's produced in an environment where dogma is questioned, rigor is enforced, and where there's deep and broad experience. We have that with the faculty here, but the part I would say that really makes us excel here, that makes the University of Montana really a better place for wildlife research than you can imagine any private think tank or private company or governmental research station having, is what I would consider to be the brilliant system of a research university. What we have here, this is a collage of some undergrads that I've been fortunate enough to be able to take into the field and to do research with. We have this amazing system at the research universities where undergraduate and graduate students are brought together with professors that both teach and research. Because professors have time to do research, their teaching isn't just a repetition of stuff that's found in textbooks. The teaching instead is alive, describing the research as it actually happens before it gets into the textbooks. And of course, then there's the stories. What happened when this research was picked up by the press, or by the Forest Service, or by the governor's office, or by Congress. Likewise, in a research university, the students aren't just passive fodder to be fed facts. Students are irreverent. They're full of questions. They're bright and unburdened by the dominant paradigm. And research professors benefit from that as well, because those ideas can get injected, and they certainly have in my career, directly back into the research. Those irreverent questions, they're awesome. And then, of course, there's graduate students. Here's just a few of my graduate students over the years. And, of course, these folks have been instrumental in much of what I've talked about tonight. The graduate program here is more competitive than virtually any academic program, including medical and law schools in the country, and our applicants come from around the world. So, quite literally, our graduate students here are the best in the world. They inspire me on a daily basis, and I guess I wanted to end by saying that I appreciate all that all of the students, have taught me. And so with that, I would thank you for your time and your attention, and I'd be happy to answer any questions.
Well, let me just thank Scott for that, and let's see if anybody, I know a few of you have other lectures to go to and things, because there's a full slate tonight, but do we have some questions from anybody here? Let's go right over here first. Here, let me see if I can get this one.
I kind of have an idea, like, why don't you raise the animals in captivity and release them back to the wild?
Okay, why don't I raise animals in captivity and release them back in the wild? It's a really good question, and we definitely do a lot of that in wildlife biology. The story that I started with at the beginning, with talking about like peregrine falcons and bald eagles that were almost extinct from DDT, the way they came back was that very hardworking people figured out how to raise them in the wild, figured out how to raise them in captivity, put them back in the wild, get the adults to accept the eggs, and that was key to the recovery of those species. So we do that a lot. We try not to raise animals in the wild unless we have to, because there are some complicating issues that come with raising them in the wild. So we'd rather not have to, or raising in captivity, we'd rather not have to bring them in captivity, but we definitely do. And so it's definitely a career option for you.
I'm right here. What was causing the foxes to move into the Olympic National Park?
Oh, the coyotes?
Or the coyotes, sorry.
Yeah, that's okay. Coyotes do what coyotes do. And a couple of things that coyotes do is reproduce. 3 things they do is reproduce, survive, and disperse. And so I think it is actually a complicated question. But coyotes tend to be a little bit like the deer mouse in that they tend to be subsidized by lots of human modifications. They tend to do very well in a human-modified landscape. And so what we've seen with coyotes is they're spreading all over the United States. And so I think they've just spread. But it is an interesting question. It's actually one we're interested in, because one alternative is that some lost coyote stumbled up into the high country of Olympic National Park, and all the coyotes up there are descendants of the one that happened to make it there. Okay, that's one option. And if that's the case, then the Park Service could go in and remove just those coyotes that are the offspring of the founding coyote, and the problem would be solved. Right? That's option 1. Option 2 is that coyotes are just pulsing onto the Olympic Peninsula, and they're just coming on a regular basis into the high country, which means that any kind of coyote reduction program would have to be long-term and sustained. And we know from experience that it's hard to reduce coyote numbers if they're pouring into a place. And so actually we're interested in using genetic tools to address this question, because there'd be a different genetic signature if coyotes had just come on the peninsula and then basically been isolated, and their offspring are the ones that are there, from a constant and continuous pulsing on of coyotes. By the way, I have to say as a disclaimer, this is one of our hare trappers, so you got to watch out for this question.
Well, in response to that question that was just asked, I was wondering, have reintroduction of wolves been considered in controlling coyote populations?
Yeah, that's an interesting one. This is, it's great to be a professor, because you can answer those kind of questions and not really worry about getting fired. If you work for people who work for government agencies, I can't answer that question. It's very controversial. Of course, Perry has to pick up after me, but he's luckily been tolerant. People have talked about that. They've said, well, why don't we reintroduce wolves into the Olympics, and then they'll get rid of the coyotes. But there has been active discussion in the park of reintroducing wolves. But the thing is that you can agree or disagree with reintroducing wolves into the Olympic Peninsula. But one thing that's pretty clear biologically is that they wouldn't have much, if any, role in reducing the coyotes there, because certainly not in the park, because the wolves there historically used the valley bottoms, and that's probably what they would do in this case. Whereas the coyotes, of course, in this case, are up in the high country. So probably what happened would be wolves would fill in the lowlands, the coyotes would stay up high. Now, it might reduce the movement of coyotes if they are continuing to pulse onto the peninsula. It might reduce their number, or it might tend to reduce that, but it wouldn't get rid of the coyotes that are up high already.
Okay, another one?
Yep.
Let's go over here and then we'll come back.
Thank you. Actually, I've got a couple of questions. What happened with the goats on Olympic?
Well, that's an interesting question. What happened was, this is actually a discouraging one for me to have to answer. After 20 years of collecting actually extraordinarily powerful scientific information showing the effect of the goats on these endemic plant species, the park, and also after about 10 years of the park going in and live removing goats, because they thought, well, we'll try to live remove them. And in doing that, they basically got rebellion from the ranger staff who was doing the captures, because the ranger staff said, look, we're having to do these one-skid landings on the edge of 800-foot drop-offs. Somebody's going to get killed doing this, and we don't want to do it anymore. And so the park considered all these things, the damage done by the goats, the difficulty of live removing them, and the park's EIS came to the conclusion that goats needed to be killed by marksmen. Well, some folks that didn't like that idea got Slade Gordon's ear in the Senate, and he basically told the park, if you start killing goats, I will see to it that you don't get a new pit toilet in that park. And so that EIS was never acted on, and there's more goats now than ever in the park. So it's one that, Patty Happy, the management biologist there, Kathy knows her, she just doesn't want to talk about it, the goats. So, yeah.
Well, my interest is in black-tailed prairie dogs, and there's a large town at the CMR refuge, and it's a perplexing situation, though, because Fish Wildlife Park has them as a dual status animal. They're either under the control of the Department of Agriculture, and if they're the controlling force, they're viewed as a pest. And if it's Fish, Wildlife, and Parks, then they're an animal of concern. Well, it's kind of a goofy situation they have there, because on half that refuge, they're shooting them, and then on the other half, they're trying to protect them because they've got black-footed ferrets in there. I think there's 9. I was at the National Conservation Center a couple of weeks ago in West Virginia, and they said that there's 9 left. Well, this is kind of a goofy setup. I just wonder what your impression was of that. I don't want you don't have to say anything bad about
Well, no, it's
the
okay.
state.
The reason I'm smiling is because I actually took my population ecology, my wildlife population ecology class there this fall, and several of these guys are here in the audience, so I was smiling because probably they could answer that question, because we spent 3 days talking to people on all sides of this issue. We talked to the guy at the Zortman store who makes his living selling supplies to people that shoot prairie dogs. We talked to Randy Matchett, the Fish and Wildlife Service biologist, who's been the main one recovering black-footed ferrets there. We talked to ranchers, and we talked to Nature Conservancy folks. And so it really is a complex issue, I guess is what I would say. I mean, this is another one of the examples I would say where the decision falls back on. Obviously, it's going to be a societal decision on what's made, and there's lots of roles to come to facilitate that. And one role is providing biological information about, for example, how much does prairie dog shooting, how much is it compensatory? How much does prairie dog shooting decrease the numbers of prairie dogs? How many prairie dogs are needed for black-footed ferrets? How many black-footed ferrets are needed to have a sustainable population? What is the interaction with plague, which, as you know, is the biggest bugaboo, and why ferrets need lots of area? So it's complicated, for sure. Yep.
I'm just curious, how do you determine that daylight was what triggered the hair color changes?
Yeah, good question. You know, actually, that's a really good point, and I guess I don't know that. We're surmising that because everything we know about circadian rhythms, so daily rhythms that animals have, and what's called circannual rhythms, yearly patterns that animals have, whether it's migration or hibernation. For places where people have gone in and done the brain physiology and done the biochemistry and looked at the pathways, it traces back to the hypothalamus, pineal gland, and day length cues that end up releasing things like melatonin that drive circannual patterns. So we're just assuming that because the molt is a circannual pattern, that it's driven by the same basic structure that drives other annual changes. But we don't yet know anything about the mechanism. I should say also temperature might matter, and so we're also putting little temperature loggers on them as well. That's another story.
Okay, I don't see any more, so what I would like to do is invite everybody over to some refreshments that are over here on this side. And so we're going to do that. Scott will still be around if you got individual questions, but let's just join with me for one more round of applause for our speaker tonight, Scott Mills.
Thank you.